Quanta Bio. Ignite a spark. Fuel more insights. The Quanta Bio SparQ RNA-Seq HMR Kit features seamless integration of ribosomal RNA and globin mRNA depletion for human, mouse, rat, or HMR samples on Illumina sequencers. The fast workflow prepares sequencer-ready libraries in a single day. The extremely simple protocol includes 3 reaction tubes, 9 steps, and 9 components. The SparQ RNA-Seq HMR Kit is intended for molecular biology applications. This product is not intended for the diagnosis, prevention, or treatment of a disease. This video shows the protocol steps for preparing 8 libraries using master mixes. Before you begin, prepare the reagents that are not included in the kit. These reagents include the 10 millimolar tris buffer, the nuclease-free water, 80% freshly prepared ethanol, and the adapters. Mix 300 microliters of the SparQ Pure Mag beads with 6 microliters of the Bead Booster for each library preparation. This mixture generates enough beads to take 1 sample through the entire library preparation process and should be kept at room temperature during the preparation process. At each step, master mixes can be prepared for multiple reactions and stored on ice until use. This first step combines RNA fragmentation, random priming, and riboglobin depletion in a single step. The required reagent for this step has a blue cap. Start by pre-programming the thermal cycler with the heated lid set to 105 degrees Celsius. Start the cycling program to preheat the block to 94 degrees Celsius and pause while you get the reaction tubes ready. First, thaw the RNA and the Frag Prime RG depletion mix on ice. Add nuclease-free water to the desired amount of input RNA to make a total volume of 6 microliters. Now add 4 microliters of the Frag Prime RG depletion mix to the RNA solution to make a total reaction volume of 10 microliters. Mix well and resume the cycling program. The first strand cDNA synthesis occurs in the same reaction tube from the previous RNA fragmentation and riboglobin depletion step. Remove the first strand enzyme mix with the brown cap from the freezer just before use and thaw on ice. Please note that nuclease-free water is needed for this step, and it is not provided as part of the kit. Pre-program the thermal cycler with the heated lid set to 75 degrees Celsius. Start the cycling program to preset the block to 25 degrees Celsius and pause. Prepare the first strand synthesis master mix by combining 4 microliters of the first strand enzyme mix with 6 microliters of the nuclease-free water for a total volume of 10 microliters. Note that the first strand enzyme mix is viscous, and care should be taken when pipetting. Then, add 10 microliters of the first strand synthesis master mix to the 10 microliters from the fragmentation and depletion steps for a total reaction volume of 20 microliters. Mix well and resume the cycling program. Briefly centrifuge the reaction and place on ice. Proceed immediately to the second strand synthesis and end polishing. The second strand synthesis, strand marking, and end polishing occurs in a single step in the same reaction tube. This step generates the directionality of the end libraries. The required reagents for this step have green caps. Thaw the second strand buffer on ice. Remove the second strand enzyme mix from the freezer just before use. Pre-program the thermal cycler with the heated lid set to 75 degrees Celsius. Start the cycling program to preset the block to 16 degrees Celsius and pause. Prepare the second strand synthesis master mix by combining 20 microliters of the second strand buffer and 10 microliters of the second strand enzyme mix, for a total of 30 microliters. Add 30 microliters of the second strand synthesis master mix to 20 microliters of the first strand synthesis product for a total reaction volume of 50 microliters. Mix well and resume the cycling program. Libraries need to be cleaned up prior to adapter ligation. The samples will be highly viscous prior to cleanup because of the sequential enzymatic reactions. Equilibrate the SparQ Pure Mag beads to room temperature for 20 minutes before use. Add 90 microliters of the beads to the ligation sample. Incubate the mixture for 5 minutes at room temperature. Pellet the beads on a magnetic stand until clear. Carefully remove and discard the supernatant. Keeping the tube on the magnetic stand, add 200 microliters of freshly prepared 80% ethanol to wash the beads. Pellet the beads on the magnetic stand for 30 seconds and then carefully remove and discard the supernatant. Repeat the wash one more time and discard the supernatant. Air dry the beads on the magnetic stand for 3 to 5 minutes, or until the beads no longer appear shiny. Take care not to over-dry the beads. Remove the tube from the magnetic stand and resuspend the dried beads by adding 68 microliters of 10 millimolar tris buffer and mix well by pipetting up and down. Incubate the beads at room temperature for 2 to 3 minutes. Pellet the beads on a magnetic stand until clear. Carefully transfer 65 microliters of the supernatant into new thin-walled PCR tubes and proceed to the adapter ligation step. Alternatively, the sample can be stored at 4 degrees Celsius for up to 24 hours. The protocol has been optimized with SparQ UDI adapters, but is also compatible with any full-length Y-shaped adapters. The adapters may need to be diluted using the UDI dilution buffer before use. Please refer to the product manual for optimal adapter ligation efficiency. The required reagents for this step have orange caps. Thaw the rapid ligation buffer on ice. Remove the T4 DNA ligase from the freezer just before use. Pre-program the thermal cycler with the heated lid off. Start the cycling program to preset the block to 20 degrees Celsius and pause. Combine 20 microliters of the rapid ligation buffer and 10 microliters of the T4 DNA ligase for a total volume of 30 microliters of the ligation master mix. Add 5 microliters of the diluted UDI to each sample from the cleanup step. Transfer 30 microliters of the ligation master mix to each tube containing the sample and the adapter for a total reaction volume of 100 microliters. Mix well and resume the cycling program. Briefly centrifuge and proceed to the post-ligation cleanup. Please refer to the post-second strand synthesis cleanup step for the detailed protocol. The bead input and resuspension volumes differ at each step. For this step, add 70 microliters of the SparQ Pure Mag beads slurry to the adapter ligation product. Wash the beads twice, each time with 200 microliters of freshly prepared 80% ethanol. After air drying the beads, resuspend the beads in 25 microliters of 10 millimolar tris buffer. Carefully transfer 23.5 microliters of the supernatant to a new thin-walled PCR tube and proceed to the library amplification step. Alternatively, the sample can be stored at 4 degrees Celsius for up to 24 hours. Thaw the Hi-Fi Plus master mix and the primer mix with white caps on ice. Pre-program the thermal cycler with the heated lid set to 105 degrees Celsius. Take note of the recommended number of PCR cycles at step 3 of the cycling program. Start the cycling program to preheat the block to 37 degrees Celsius and pause. Prepare the PCR master mix by combining 25 microliters of the Hi-Fi Plus master mix and 1.5 microliters of the primer mix for a total volume of 26.5 microliters. Add it to the 23.5 microliters of the purified adapter ligated DNA for a total reaction volume of 50 microliters. Mix well and resume the cycling program. Briefly centrifuge and proceed to the post-amplification cleanup step. Alternatively, the sample can be stored at 4 degrees Celsius for up to 24 hours. Please refer to the post-second strand synthesis cleanup step for detailed protocol. The bead input and resuspension volumes differ at this step. It is important to note that there are 2 bead cleanups within this step. Perform the first bead cleanup by adding 45 microliters of the SparQ Pure Mag beads to the library amplification product. Wash the beads twice, each time with 200 microliters of freshly prepared 80% ethanol. After air drying the beads, resuspend the beads in 52 microliters of the 10 millimolar tris buffer. Carefully transfer 50 microliters of the supernatant into a new tube. Repeat the bead cleanup step by adding 45 microliters of the SparQ Pure Mag beads. When resuspending the dried beads for the second time, add 32 microliters of the 10 millimolar tris buffer. Then, carefully transfer 30 microliters of the supernatant into a new tube. The sample can be stored at 4 degrees Celsius for up to 24 hours, or at -20 degrees Celsius for longer-term storage. Proceed to the library QC step. One option is the qPCR-based quantification with the SparQ Universal Library Quant Kit. The libraries are now ready to be sequenced. Quanta Bio. Ignite a spark. Fuel more insights.